My name is Anaki Effendi, and my summer research at the LMSE lab has centered around computational protein design. I will present a Gaussian process regressor-based machine learning model for phosphatase activity prediction. The working model I made can be found on GitHub. The aim of my research was to develop a machine learning model that when fed a protein sequence for a given substrate would be able to predict what the protein activity would be, Vmax if you will. The machine learning model I will be presenting today uses a Gaussian process regressor approach to perform this prediction. The significance of this research is such that it allows for the faster identification of viable protein sequences. Typically, researchers will spend months performing high-throughput screening, sampling on almost every variable in a theoretically infinite search space when trying to identify and optimize designer proteins. With this machine learning model, researchers will be able to identify viable protein sequences in a shorter amount of time using a targeted constrained search space. These proteins have many applications, ranging from drug discovery and pharmaceuticals to use in sustainable technologies such as bioreactors to the production of fermented foods. As I mentioned earlier, my model uses the Gaussian process regressor, GPR. A GPR is a multidimensional regression method which allows for curve fitting. As you can see from this diagram, the solid green line represents the mean of the observations, while the gray region represents a 2 standard deviation uncertainty around the curve. Essentially, a GPR produces entire families of curves, where there exists a nearly infinite combination of curves who all exist within a certain margin of error. As you can see from this blue line, we can then obtain predictions using this model of what the output would be on an unseen, untested input. The probability density function of the fitness of the sequence can be seen by this red line and is normally distributed. The GPR allows us to focus and constrain our search space on regions which have a higher probability of yielding more desirable proteins. The shape of the curve is determined by a covariance function, also known as a kernel function, whose shape is in turn governed by a set of hyperparameters. For the purpose of this investigation, I had settled on the Matern kernel, as this is what is used most commonly in literature. During times like this, we are not able to access the lab to conduct experiments ourselves, and so we had to source our data elsewhere. We used protein sequences and protein activity data from a paper which had investigated the haloalkanoic acid dehalogenase (HAD) superfamily. HAD are a superfamily of phosphatase proteins. This paper tested 218 different phosphatase proteins on 168 different substrates to measure their activities. The first diagram illustrates a map of the substrates that were on the test plate. The second image is the activity data of just one protein on these substrates. I used a Python script to extract the sequence data from NTBI and UniProt protein databases. Having a large dataset like this to train and test machine learning models is key to making a good machine learning model. Before we can train and test our model, there are a few important data encoding and preparation steps we have to perform. As you can see from this histogram of the protein sequence length, most of the sequences were shorter than 300 amino acids in length, and so we cut out the sequences longer than 300 in length from our dataset. When we trained and tested our model with these long sequences, it performed poorly as it was not able to generalize well. Next, we performed a sequence alignment on the protein sequences using Clustal Omega, which is the current state-of-the-art protein sequence alignment software and method. This sequence alignment step is important as it allows for two things. Firstly, the aligned sequences are all the same length, and this makes it easy to feed into the machine learning model, which requires all input data to be the same length. Secondly, the aligned sequences are easier for the machine learning model to identify features from and group trends of amino acids within the sequence so that it can attribute to the resulting activity. Lastly, we performed the encoding step. We chose one-hot encoding as the encoding method of choice, as it allows for the simple conversion of the protein sequence into a numerical matrix format. This step is crucial as the machine learning model cannot perform logic on strings. It can only process them numerically. I will briefly explain how the method of one-hot encoding works. If we have a given dataset of two sequences, we can start by encoding just the first sequence. Firstly, we'll perform a padding step using the dash symbol to fill in the spaces so that all the sequences are as long as the longest sequence. This step is already done by Clustal in our case. Secondly, we couple the padding sequence with an amino acid lookup table and perform a numerical indexing on the values each IUPAC amino acid was assigned. Lastly, we perform a vectorization step, which represents each entry in the sequence as a column of vectors of zeros and a 1 in the row corresponding to the indexed amino acid. In order to measure the performance of our model without having to collect more data, we came up with a way to perform a train test loop using the same dataset. Firstly, we randomly shuffle the dataset. Next, we split the data, 80% into a training set and 20% into a test set. Then we feed the training data into a GPR hyperparameter optimizer, which optimizes the hyperparameters on the kernel function to the training set. These optimized hyperparameters are fed into a regressor prediction model along with the protein sequences, X if you will, from the test set and produce a prediction of the protein activity, Y. We can compare the predicted activity from the test set to the real activity. This is plotted on the diagram below for the substrate L-arabidyl 5-phosphate, and we can measure the performance of this model on the substrate using the correlation coefficient R. This split train test loop was performed about 20 times for each substrate for efficacy of results. This final experiment took about 6 hours to run. As you can see from the diagram below, out of the 168 substrate models made, the model was somewhat reliable on 19 of the substrates and had a decent prediction on 16, while the remaining 133 substrates had almost no correlation between the predicted and measured activities. When developing future models, we aim to move our model performance up the line and into the green zone of both good model prediction and consistent prediction. The poor overall performance of the models can be attributed mainly to a few factors. Firstly, we only tested with the Matern kernel so far, but there do exist other kernels which could provide better results. Secondly, GPR is not the best model for sequential data, as it is a simple model, and when trained on such high-dimensional data, it does not generalize well on unseen data. Lastly, the data set we have is quite small, and when compared to the length of the sequences we have, it is not adequate. Hence, more hand engineering and thus more layers should be used in the model. In this case, we only used one. In the future, we will explore GPUCB, which is a Gaussian process model which performs unconstrained bandit setting to provide better recommendations with minimal regret. Adding more layers such as one-dimensional convolutional layers and testing other methods of encoding such as natural language processing will improve the models. Despite the not-so-stellar performance of the models, this project was successful in demonstrating a baseline machine learning model to perform some feature extraction and prediction capabilities. I would like to give thanks to Professor Krishna Mahadevan for allowing me to work in his lab, Paul Zhu for being a great mentor, Kaushik Raj for being a great mentor, and Chester Pham for helping me out with this project.